Um, I guess I can just introduce myself. Yeah, so nice to meet you, everyone. And again, I got this heavy duty of being here and making sure everything was going well. So thank you for your patience. My name is Bora Lim. I am Associate Professor in Baylor College of Medicine. And I wish that the title was actually Meeting at the Bar after this session of the technical difficulties. But we would like to share that, when is a good time, as a clinical translational researcher, when is a good time that you have a discovery that can come into the clinic. So these are my disclosures as a clinician that we work with a lot of companies in various settings. So I would like to start by the question that everybody knows, that why is it so important for us to translate and do this kind of sessions and workshop with everybody. Every October we do walks and sessions. And that's because we lose a woman and sometimes men every 12 to 13 minutes to breast cancer. So during these 2 hours that we had a wonderful talk, that we have lost number of women to breast cancer as of 2021. So how are we going to change that, and what are we going to do to change that? That's why it's so important for us to meet and talk about the translational research. So I wanted to kind of highlight, this is a kind of like notification from the approved FDA drugs in 2020. If you remember last year, we were just starting to learn about the COVID. We lost a lot of lives there. If you're looking at the older list, there's about 64 that had been approved by the FDA in oncology. If you're looking into 2021, despite all the COVID and all the challenges that we have experienced, I'm counting 115 and more that is approved by the FDA in clinical oncology as benefiting patients today. So it's very, very exciting for a person who's doing the translational research in cancer. And I think that's why we are all gathering here, despite all the challenges and Omicrons and so on and so forth. So the next question is, okay, so we understand that why it's important, why we are gathering here and doing all these things, but the question is, what are you going to translate, So we had heard a wonderful talk from Dan and, Jen and Senthil. We went over all this gene editing-based research, how you're going to screen the in silico data, trying to discover all these important gene targets, as well as how you're going to validate that or test that in the, patient-specific models even. So for a long period of time, this is just one example in the inflammatory breast cancer, where we started with the microarray and then went into the hotspot mutation and the ampli-seq, now going into the era of next-generation sequencing, whole exome sequencing, and even comparing based on the clinical response, what are the potential genes that are differentially expressed based on the response versus non-response. And by doing so, we are really trying to understand. So this is something we call clinical specimen-based research. So, as many of you know, that the Human Breast Cell Atlas is a part of the big Atlas project. We think that it's very also important to note that there's a new technology that we are trying to utilize, but we also should not forget that there's always negative control. So when you're studying the cancer, a lot of times that I learned through this Human Breast Cell Atlas project that I'm doing with my colleagues is that actually a lot of things that we think that is only cancer specific, like for example, fibroblast or macrophage, we actually find that in the normal breast cell as well. So it's very important to kind of ground ourselves and really learning by really studying the clinical samples. You really have a lot of efforts ongoing with the understanding tumor microenvironment. Now we're at the point that we can actually study them by creating an organoids model, but we have to start from somewhere by actually implementing information from the studying patient samples. By doing so, we can have a targeted potential pathways that we can find as a vulnerability, and then we can target them in the clinic. So when you have this ideas and go through the in silico, so for example, GRAB7 is one of the important targets. We know that it's included in the oncotype. You're developing a good models to study this. Then, okay, so when you have a target, if you're a medicinal chemist, of course, you can develop your own drug. But if there's any good drugs out there, it's okay to go ahead and study them as well. So this is actually one of the examples that, kind of it's going in the backwards, that a lot of times that we feel that we need to know what's the target that we're going to start from there and then further develop through the preclinical models. But there are some other ways that by communicating among the translational team, other ways you can actually develop a new targets or potential tumor microenvironment cells that you're going to move on to the next mode. Is that, for example, this is one of the clinical trial that the Baylor cellular and gene therapy colleague is doing. So So what they're doing is the oncolytic virus that is invigorating the IL-12 as well as the usual GCSF by attracting both systemic and local therapy. And then actually the patients were supposed to get the HER2 targeted CAR T, but initial virus only modification patients will only receive the virus only. So this is one of my patients. So this patient has an ER/PR positive disease where a lot of us are thinking that, oh, it's an immune cold tumor. But this lady has progressed on a lot of different medications. When she finally decided, okay, this might be a good, item for me to try, she had this huge neck mass that it was pressing her nerves, so she couldn't really lift her arm. She had an Orner syndrome, and so on and so forth. So we injected the virus, after reviewing all of her case, and everybody approved. And then initially there was some increasing of the mass by the virus activity. She had a fever and all the symptoms. By day 21, actually, we decided that we're going to actually give the 1 dose of pembrolizumab, despite this is not the really targeted drug for the ER-positive tumors. And by day 42, her tumor has almost regressed and disappeared. So what did we learn from her peripheral blood after injecting all of this CADVAC? So this is a calendar, how her blood was collected in each time point. So we have learned some few things. Her tissue is currently undergoing the analysis, but even by her studying her peripheral blood, we realized that actually there's a transient reduction in the pink that is disappearing by day 4 completely from her peripheral blood, which is later actually showing up back again in her peripheral blood. How about some of the other T cells? Actually, the patients had an increase of the activated CD8 T cells and Tregs over time, even all the way up to 4 weeks period of time. And compared to some of the other patients who had been on this study, we think that this might be a potential secret that, she's responding so beautifully. So expanding this idea that we are also collaborating with the other PhDs and MDs like of our team, what they also have found out is that there, in addition to the changes of the NK cell and T cell proximity, we also notice that there's a wall of fibroblasts surrounding luminal B cells in this patient's tumor, potentially inducing there's a limitation of how this T cell can actually approach. So even with the pembrolizumab, without breaking the barrier, that we can really not approach that sample. So therefore, I'm encouraging my PhD friends that, hey, this CADVAC3 is not enough. We have to develop a K-tetra. In addition to having the oncolytic virus, we also have to remove these irrelevant T cells and inducing the T cell that is relevant to the tumor. So we are actually actively studying this pathway by also developing models and further virus. And of course, as a translational researcher, that when I talk to my colleagues in the basic science, of course, identifying the initial target is very, very exciting. But at the same time, it is also important for you to understand that emerging resistance could be very important. So many of some of the investigators I talk to say, hey, Bora, I have this wonderful drug. I do the animal study and that is actually responding. But as you realize that many of these patients initially respond to the treatment, but quickly actually develop an emerging resistance. Therefore, understanding where is actually potential of these new clones or heterogeneity of the clone and new mutations by pressuring by the therapeutics, understanding that aspect it can give us a lot of insight into vulnerability. So for example, this is how we think about the drug development as a clinical translation researcher. So for example, there's so many things that we can actually do for this patient. This patient, let's say, started from the very high lymph node. We know that this is very high risk for the metastatic disease. There is a possibility that you can actually induce a beautiful response in upfront during the neoadjuvant therapy, and then validate all these wonderful RCBs that you have heard throughout the talk and then session. But if there is still residual disease, maybe we can do the biopsy and understand what's the emerging resistance there. And by doing the window trials, maybe we can understand a better outcome. Once the patients go through the local therapy, we have another opportunity, maybe we can intervene. So you have seen that TNBC, for example, there had been a huge change in the adjuvant setting from where we didn't have any adjuvant therapy options into now there's a multiple therapy options, depends on the BRCA proficiencies and deficiency, some of so on and so forth, and the other factors. So just to kind of get the glimpse of what's actually available out there in the clinical setting is also important. How do you know that your either drug or pathway, biomarker, your target is ready? Of course, in addition to having in silico screening and understanding, of course, you have to have a rationale. And I think largely it comes from the really talking to your colleagues and the thorough literature search. You have to have a mechanism. It doesn't have to be right, but you have to understand why, if I were to target this target and then make a difference in the patient sample tomorrow, then what's the mechanism that I'm going to go after? And of course, there is a wonderful talk that previously my colleagues have done, so I don't really need to go into any of the details. But the other thing I want you to pay attention, no matter where you stand in your spectrum of translational research, is of course pay attention to what are the available molecules. In addition to wonderful TKIs, now we have ADC, there's a PROTAC, there's a lot of like this Optimer, Aptamer. There is a developed form of the developmental therapeutics that we can use. Now, I'm going to share a really brief 2 stories on the translation because, like, not that I have been doing this for too long, and I'm sure many of you in the audience have done this longer than I have and have done wonderful things, but, I just wanted to kind of share, like, it's not always rosy and not always a success story. So there was a time during my fellowship that I had been starting to pay attention to the MCL1. So at the time that my focus of the disease subtype was a triple-negative breast cancer, and there had been a report by the Balco et al. In the Vanderbilt group that once you have a patient either compared to primary to the metastatic, or have a patient who didn't really respond and clear their tumor after the 6 months of neoadjuvant therapy, it seems to be that MCL1 is one of the high amplified genes or protein that maybe that's a good target for you to overcome the resistance if you have those MCL1. So there had to be a better way to target the MCL1. As many of you may remember, there had not been a good MCL1 targeted therapies at the time. This was about 7-8 years ago. At the time in the Ueno laboratory that I belonged to, that we had a wonderful Angie's, the PhD students, and Jason, who was actually mentoring her in the experimental, we were actually screening this, the TNBC cells randomly with the multiple drugs of the interest. And during that time that we have discovered if you have a entinostat, the HDAC inhibitor we heard previously in the plenary session, if you have a different resistance versus sensitivity to the entinostat, actually the resistance could be through the NOXA, which is the comprehensive, the combining molecule of the MCL1. So we further developed the study by screening and combining the entinostat with the other the potential combination partner. What we have discovered is that actually, if you're combining the MEK inhibitor through the RAS, the MEK kinase pathways, but also it has a peculiar activity of the modulating some of those, the MCL1 mediated, you could actually eradicate some of the MCL1 by combining the NOXA. So if you're actually treating them with the combination of the entinostat and then the MEK inhibitor, you could actually induce the eradication of the MCL1, thereby you could actually induce the good response in the preclinical model. We confirmed that by using the animal model at the time that we used the pimasertib, but we also have tested in the multiple MEK inhibitor, confirmed in the TNBC models in those potentially the NOXA or the MCL1 becoming the resistant mechanism, you could actually induce the response by combining these two. So by developing this, we have also developed multiple biomarkers that, because, as you can imagine, MCL1 is not an easy target for you to test. There is really no strong, robust IHC that is available because MCL1 and NOXA has to complementarily bind. So we had to develop a heterodimer detection mechanism with the IHC. We're using the RNA-seq. We got the RPPA gathered around. We spent a long time develop this model and finally got this MCL1 degradation by using the entinostat and then MEK inhibitor combination approved by the CTAP to move forward to the next clinical trial. But unfortunately, at the time that we were launching this trial, there was no available MEK inhibitor in the CTAP, and therefore, after 3.5 years of work, we had to actually completely demolish the project. I mean, there's still my colleagues that are continuing to develop this, project, but it was really devastating. It was part of my grants and all these things, and we had a patient who were waiting for this the clinical trial to launch, and it was sad, but no experiences go wasted because we learned so much about how this MEK inhibitor can actually have other activities and so on and so forth. When we had other opportunity to relive this binimetinib, which is another MEK inhibitor, it's out there, we took the opportunity, and then now we have another NCI match study that is upcoming. So some of you may know this already, but NF1 is a well-known target of the melanoma that is inducing the RAS-MAP kinase pathways related aberrations. But more importantly, in the ER-positive breast cancer, we also note if you have a certain missense for the frame shift mutation, you could actually shift ER-positive cells into more on the dependence in the RAS-gap pathways. And then if this NF1 mutation is very rare in the primary, but in the metastatic setting, it actually goes up. It actually does not eradicate completely of dependency or the response to the SERT, but if you treat them long enough, eventually they will develop a SERT activity. If you're treating them with tamoxifen or the aromatase inhibitor, they're not really sensitive at all. We had an expert in the lab who was studying this, the NF1, the whole gene sequencing. We actually found out there's a two different binding domain, not only directly interacting with the RAS-gap pathways, but also directly interacting the E2, the estrogen receptor. Thereby, if you do have a certain mutations that could actually hyperactivate it in the RAS-gap interacting, in addition to the ESR1 that is interfering with the estrogen binding and then therefore developing resistance. When we tested in the animal model, this is one of those, the PDX model that we have developed in the Baylor College of Medicine. As you can, it's a very complex, the graph, but basically, if you let them grow, cancers, of course, will grow. If you treat them with the fulvestrant alone, there might be some kind of like slowing of the growth of the tumor. But once you have the binimetinib, which is the MEK inhibitor, is introduced in the middle, then you can actually see that the growth curve will quickly, goes down. And if you have a combination in the late binimetinib combination, if you actually stop that combination treatment, that tumor, again, following the initial growth curve and growing back again. If you continue the fulvestrant at the binimetinib combination, even at the really later in the 231 days, there's some continuation of the combination. In some models, we also have found that even after we stop the binimetinib, this tumor growth inhibition is actually persisting. So utilizing this time and then our knowledge about the MEK inhibitor, this time we didn't miss the opportunity. We were able to get the clinical trials as a combo match. There's a 2-arm that we're going to study that patients who had not been exposed to fulvestrant, we're going to give them in the randomized fashion. If you had been exposed to fulvestrant, we're going to give them in the one combination. We're going to have a deep dive and understanding that who might be actually respond better. Because it's the other thing that we have to kind of keep in mind, that translating all of this cool idea is that you really need to know who's going to benefit because, like, for example, this young lady is trying to go for a dinner party and she's picking out the shoes. I mean, this is not even relevant, compared to cancer. But if you are trying to treat the cancer, you really, really need to know what cancer you're going to treat because many of you know that breast cancer is a very heterogeneous group and very complex. So even at the early on, when you're doing the cell line study and then PDX study, PDO study, if you really know who's going to be your target group, that gives you a huge advantage on translating your ideas. And one of the reasons is because sometimes that we are targeting the loss of the NF1 by the functional upregulation of the MEP kinase pathway, and yet your usual RNA sequencing may really not correlate with your NF1 true loss in the level. So right now, we're working really hard and trying to figure out if there's any IHC or the KIP or the proteomics-based data we can actually develop and trying to figure out who might be the person who's going to respond. So those work is currently ongoing. There's some of the other complex aspect, for you to kind of keep in mind, and you may say, hey, Bora, I am kind of barely making with this awesome drug, how you're gonna really think about that. Of course, this may not have to be your job, but if you feel like you don't want to think about it, at least get those people on board on your team. And this is for example, the commonality of the discussion. What is the complementarity of your biology? Are you going for the multiple targeted pathways based on your therapeutics? And then what is the process? If you have a target, you're going to try to identify, and then now you develop the drugs. What is your prioritization strategy? What is your development strategy? And then if you have already available inhibitors, do you have a means to test them? How can you say that your drug is better than the other drug? And of course, it's always important to have a multiple expertise and get them in the very strong organizations that you can actually move the steps forward despite how complex your ideas might be. You can also decide how far you're going to go. These are the steps that, it's very basic steps for your drug. And, I just wanted to point out that I'm just focusing on therapeutics because the other talks were covered by my other colleagues, that there's so many steps that for your drugs from the cell line study, animal study, GLP study, phase 1, phase 2, phase 3, multiple steps that you have to go through, but you can decide. You can be an awesome quarterback who kind of saw the opportunity and then just grab the ball and just run all the way to the touchdown. Or you could actually work with your team and say, hey, I have done this much. now is the time for me to pass on to my colleagues and then further develop. So whoever does the next work, as long as it hits the clinic, benefit the patient, we have done our job. When is a good time to talk to your translational colleague? And translational colleague that I mean here is by somebody like me or somebody who has done more other type of experiment in this field. So you make sure your idea actually is able to be translated. So when is really a good time to talk about this translation of your idea? What molecular target is ready for the prime time? Involve the translational colleagues, when is a good time? I say as early as possible. I mean, this is a conference. We used to have a lot more people, and, there's a very complex kind of like networking sessions and so on and so forth. But this might be a good opportunity for you to reach out to somebody and say, hey, I have this wonderful target that I'm trying to develop, and I'm using this experimental design. What do you think about that? There are so many experts out there now. You can reach out to social network and other platforms. So make sure you involve your colleagues as soon as possible, as early as possible. And when you're trying to talk to oncologists like me and many of you in the audience, this is what we go through our head. So it's like, okay, well, so the drug is amazing, but is it best used in metastatic? Is it best to use in neoadjuvant? Is it best to use in the adjuvant? Is it a window of opportunity trials? Do we need some additional data for us to move forward in the clinical trials? And so, you may think that you may know, but maybe the field is also changing. So I think it's a good idea to talk to the clinical colleagues early on. Once you have the opportunity to test your strategy, try to get as much information as possible. Always be ready to learn and improve. That's what I try to thrive and have this wonderful session. And I think one of the key success that I have seen from my colleagues who has done a wonderful job of translation is that there's really no reason to say that there is no stupid questions. So don't be afraid to ask questions and always be ready to be disappointed. Somebody will tell you like your drug is not going to work or your target is not going to work, but as long as you have a strong rationale and belief and then have a true expertise to develop, then I think there is no disappointment. And then lastly, I wanted to finish out with, like when we are trying to do this translation of research, you hear about this new target or the drug that you never heard of. And people will say, oh, you can do all this literature search. I've never heard of this. There's something we call the collective intelligence or collective wisdom, but how can you say that yours is good? But like Bertrand Russell said, actually, you might be the only one who truly have discovered something new, and maybe you can educate all of your colleagues and move on to the next step. And maybe 5 years from now, we will hear about that, San Antonio Breast Cancer Conference. So if your idea is very, very new, just to make sure that you do the literature search and like a lot of talk with your colleagues, make sure it's not crazy, but actually you may be very close to something truth. So I wanted to kind of finish up from there. Thank you for my colleagues. And if you have any questions, those are the emails, and I'm also reachable to the Twitter. And thank you for your patience. Okay. I am not seeing any questions on okay. So Alana is asking, because breast cancer.
Talk, Bora. I put a question in the chat because I know there's like a 17-second delay. So I thought that's the best way to get it to you.
Yeah, thank you, Alana. So the question is, because the breast cancer therapies are getting more complicated and multiple combinations are approved, how do you recommend people to try the model more resistant or the relevant system? Do you need to start by doing triple therapies in mice or focus on the PDX-resistant tumor? Oh my God, I think that's a wonderful question. So if you talk to like some of my colleagues, especially not necessarily in the breast, but other in the GU and GI, they will tell you this is time for the triple therapies. But we all know that, for example, your mice may not survive in triple therapies. Even like doublet is sometimes really hard to tolerate. So I think one of the things, of course, if you do it in the smaller dose, maybe there is a way that you can actually test all of those triple combinations. But I think one of the strategies that we're thinking as a better strategy is if there is a good rationale of combining, having the strong mechanistic underlying theory, then you can actually have a even doublet, and then you can even test it in the sequential. And then by doing that, we can overcome some of the limitation of the toxicities and so on and so forth. But I don't think there's any strong way. I don't know, maybe we can use Senthil's PDO, and then that way we can have a less concern with the toxicity. But that is certainly a struggle.
Bora, I was also going to ask the sort of idea of the virtuous cycle of, a discovery leading to preclinical data, leading to a trial, and then sort of the follow on from there in terms of thinking about how to use biological samples that patients donate when they're on clinical trials optimally, and how that partnership back from the trial to the laboratory can be sort of optimized to ensure we learn the most from these studies and then are able to make the next moves from there.
Yeah, then thank you for a wonderful question. So I think, so even a few years back when we used to have more traditional preclinical model where we were focusing on more about kinase pathways or the gene targets, I think there was a huge delay like we are experiencing in Zoom today from the time that somebody donated their tissue, from getting the data back, and then have some relevant ideas generated from there. But I'm fascinated that especially working in my colleagues with the cellular and gene therapy. So we had this patient that I showed you as a data, literally got treated in like September sometime. And then we have gotten some of her data back. And then by accumulating a few more patients' data and then having this wonderful collective models from my colleagues, we're actually able to bring it back and generate that additional virus. And the same patient might go on the other trials by doing that. So I think that life cycle has narrowed down so much because of the wonderful scientists like I had an opportunity, fortune to work with today. So I am optimistic that gap might be much narrower than we think. And then I think in the other way, if we expand our network and don't have to have everything on your own, I think that's another solution. Maybe we can bring it together. But yeah. There is a question in the chat box that how do you evaluate the reliability and IgC assay for tumor suppressor gene in clinical? Oh my gosh, genius questions. So for example, the NF1 is also a tumor suppressor like a P10. So I share the sentiment with you. It's very, very difficult. So in our team right now, what we are doing is we are actually having this very highly sensitive, the nanobeads tied kit, and trying to actually do the mass spec-based protein level and trying to compare them with the IgC, because we think that might be the only way we can validate. There is a long history of the P10 measurement in the IgC, so I'm also talking to the colleagues who went through the same battle like we have. So that's one of the things that we're trying to do, but I will update you once we have the solution. All right, Alana, I think we are almost up. So I'll hand back to the regional moderator, Dr. Wem. Any closing statement?
Well, I just want to say thank you all very much for attending, and a special thanks to all four of our presenters for wonderful talks. I really enjoyed the session and I learned a lot, and I hope all of you learned a lot too and are motivated to go back to your clinics or your labs and work together as teams, because I think that's what we really heard here is going to be the next step in translational science. So thank you all for attending. I hope you enjoy the rest of the conference, and hope to see you around. Sorry again for the technical difficulties as well.